Namaskar दोस्तों, welcome to channel Toxicology on Board. Today in this video we'll be discussing another important topic, that is the treatment of cyanide poisoning, nitrite mechanism of action that we'll be seeing. So let's directly dive into the question. This question is taken from ABT recertification paper only. The question is, in the treatment of cyanide poisoning, nitrites act by. So we need to identify the mechanism by which this nitrite used as an antidote for the cyanide poisoning. So we have option A, that is displacing cyanide from cytochrome oxidase. We have option B, that is oxidizing the iron in cytochrome oxidase from Fe plus to Fe plus 3. So iron exists in plus 2 state, ferric state or ferrous state, Fe plus 2 as well as Fe plus 3 state, oxidizing cyanide to thiocyanide, oxidizing the iron in hemoglobin form from Fe plus 2 Fe plus 3, displacing cyanide from the cytochrome P450. The right answer will be in the green text, which is nothing but option D, that is oxidizing the iron in hemoglobin from Fe plus 2 Fe plus 3. So let's understand the mechanism in detail. If you look into in another slide, I will explain you how the cyanide poisoning the mechanism, how it is. First, we'll try to understand the right answer, that is sodium nitrite. If you look into sodium nitrite, nitrites like amyl nitrite or sodium nitrite, how the mechanism is that whenever there is a cyanide poisoning in the case of patients, what it will do, the sodium nitrite, it will cause formation of methemoglobin. So we have hemoglobin. So whenever you are administered this sodium nitrite, it will form methemoglobin, which act as an alternating binding site for the cyanide ion. So the cyanide ion, instead of going and binding to iron in the case of hemoglobin, it will go and bind to iron in the case of methemoglobin. Okay, so once again I will try to say, in the treatment of cyanide poisoning, nitrites, amyl nitrite, sodium nitrite, they will primarily act by oxidizing the iron in hemoglobin to induce a state of methemoglobin. So administration of this nitrates, either amyl nitrite or sodium nitrite, it will induce a state called methemoglobinia. Okay, this is a typical structure of amyl nitrite, sodium nitrite and cyanide. And cyanide is a neutral molecule that you have to understand. So we have hemoglobin, methemoglobin state is induced in your body whenever nitrates are administered as an antidote for the case of cyanide poisoning. Okay, so let's see what is happening in the case of normal functioning of cytochrome oxidase and what is happening in the case of cyanide poisoning. And let us first understand, before going into the treatment, let us first understand what is how the mechanism of cyanide poisoning is there. So in the case of normal cells, what is happening, whatever this oxygen is there, no, it will go and bind with Fe plus 2 state in the case of heme group. Okay, this is your and all this action is taking place in the case of inner mitochondrial membrane. So mitochondria is the place where ATP is generated. It's the powerhouse of the cell. In that also, particularly inner membrane, in that inner membrane, we have complexes. Cytochrome 1, 2, 3, 4 is there. In that also, in cytochrome 4 complex, this particular case, this particular mechanism is happening. So in the case of cytochrome 4 complex, we have heme group in which Fe plus 2 is there. Okay, this is your cytochrome oxidase. Here green color structure you can see, this is your heme group in which in the heme group it is in Fe plus 2 state. Oxygen will go and bind to this Fe plus 2. Okay, this is what oxygen bonded to Fe plus 2. So what is happening within this cytochrome oxidase? As a result, what will happen, ATP is generated. Oxygen, whatever the molecular oxygen is there, no, it will be reduced to water molecule as well as ATP is generated. Okay, so this is what happening. Blood is there, blood is bringing oxygen in this mitochondria, particularly cytochrome oxidase, heme group Fe plus 2 is there, where reduction is taking place in, and this molecular oxygen is converted into water molecule as well as ATP. So this is a normal functioning in the case of cytochrome oxidase. What is happening in the case of when cyanide poisoning is there? Whenever this cyanide is ingested, whenever the cyanide is reaches the systemic circulation, what is happening? Cyanide will go and bind to the same heme group. Okay, instead of oxygen, your cyanide will go and bind to this heme group. This is your heme group, and as a result, oxygen is blocked from binding. Oxygen can no longer bind to this heme group. Okay, in Fe plus 2 state. As a result, what is happening? ATP production will be decreased. Okay, oxygen is not reduced, interrupting ATP production. As a result, lactic acidosis takes place. Okay, tissue hypoxia happens. Those cells which are in high demand for ATP, like your CNS as well as your heart, they will be deprived of oxygen. Then there is a state called tissue hypoxia state. It is called. As a result, the cells will undergo death. So once again, we'll see the cyanide ion. It will bind to Fe+2 center within the protein cytochrome oxidase. This is your cytochrome oxidase. In this protein, you have Fe+2. Cytochrome oxidase uses electrons from this oxidation of fuel molecules to reduce this molecular oxygen is there, no O2 to water molecule as a part of aerobic pathway. So now because of cyanide poisoning, this aerobic pathway has to be moved towards anaerobic pathway that produces ATP. Blocking this protein stops production of ATP. So ATP production is decreased, making cyanide lethal at even at low doses. This is the basic mechanism. Now we have to see how nitrite or other antidotes can be used in order to interrupt this particular toxic mechanism. If you look into the sites of linkages to the protein, this is the typical structure of the heme group in which cyanide will go and bind to this Fe, okay, iron. Okay, so as I told you, you have complexes 1, 2, 3, 4 is there. Particularly in complex 4, this particular thing is happening. Cyanide will undergo in complex 4 cytochrome C oxidase. In that also AA is the one to which it will bind. Okay, as a result, what is happening? Inactivates the cyanide will inactivate this mitochondrial oxidative phosphorylation, and it will inhibit the cellular respiration. Aerobic respiration way of pathway will slowly turn toward anaerobic metabolism. As a result, lactic acid concentration in the body will increase. Okay, so another thing you can use in case of instead of nitrates, that I will tell you. Let's first understand the mechanism of nitrates, which in 4 steps. So formation of methemoglobin. Nitrite will convert this Fe+2 in normal hemoglobin into Fe+3. Normal it is Fe+2, but in case of methemoglobin, you are converting into Fe+3, forming methemoglobin. Okay, yeah, false condition will be created in your body. Cyanide scavenging. Cyanide has an extremely high affinity for this Fe+3. So if you have cyanide and you have Fe+2 in hemoglobin, Fe+3 in methemoglobin, cyanide will have more bound towards Fe+3 in methemoglobin. Found in methemoglobin, it preferentially binds to methemoglobin over ferric iron in cytochrome C oxidase, which is the key enzyme in the case of mitochondrial respiration. Okay, so third step is reactivation of this cellular respiration. By binding this cyanide, by binding to this methemoglobin, it will form cyanomethemoglobin. Cyanide is sequestered away from the mitochondria. Okay, this allows this inhibited whatever the cytochrome C which has been inhibited to dissociate from the cyanide and resume back to aerobic metabolism. Whatever the anaerobic was there, it will come back to aerobic. Nitric oxide generation. There is another new research is taking place where they have found some evidence that stating that these nitrites will also generate this nitric oxide. So what is the role of this nitric oxide? It will directly displace cyanide from the binding site on the cytochrome C oxidase, facilitating faster reactivation of cellular respiration than methemoglobin formation. So this is another way of antidote mechanism. And next to vasodilation. Since nitrites are potent vasodilators, in the case of cyanide poisoning, what is happening? There is a vasoconstriction. By administration of these nitrates, they act as a vasodilators. As a result, what will happen? May increase blood flow to the organs, particularly liver. And you have to understand this cyanide, no, it is ubiquitously present in the environment, in the plant sources, even in small quantities available in our body. So liver and there are certain organs which have detoxification mechanism can take place in smaller quantities. So by using this as a potent vasodilator, in the case of liver, it will help in increasing the detoxification. So apart from nitrite, that is cyano kit is there. Another cyanocobalamin kit is also there for the case of cyanide poisoning, in which vitamin B12, that is hydrocobalamin, will be used as an antidote for cyanide poisoning. Okay, that was something we'll read towards the end of the presentation. Then we have amyl nitrate, which is inducing methemoglobin. Okay, sodium thiosulfate, another adjuvant you can be used for the treatment of the cyanide poisoning is sodium thiosulfate. What it will do is it will convert the cyanide to thiocyanide, and this thiocyanide can be rapidly excreted out of your body. This is the three methods. So nitrates you can use, hydroxycobalamin you can use, and sodium thiosulfate you can use. But you have to understand that all these, any type of remedy you are using, uh, it has to be used at a very rapid action need to be taken. But nowadays the first choice is hydroxycobalamin, because it has less adverse effects when compared to nitrates. What they will do, they'll create a condition, hypotension, decrease in blood pressure also they will create. So it is very challenging to treat the cyanide poisoning with nitrates. And so, but much more welcoming is hydroxycobalamin. That we will see what are the advantages when you're using hydroxycobalamin as an antidote for cyanide poisoning. So, if you're coming to clinical context, nitrates are typically administered as a part of multi-stage. This is a cyanide antidote kit will be there, okay, which is consist of sodium followed by sodium thiosulfate, which converts cyanomethemoglobin into less toxic thiocyanide. As I told you, this thiocyanide will be excreted out of the kidneys. Okay, because methemoglobin cannot carry oxygen, nitrite therapy is relatively contraindicated in patients with carbon monoxide poisoning. You have to also understand this. One of the other major causes of the cyanide poisoning is whenever there is a fire, accident takes place. No, there, because of the burning of plastics or certain components are there, because of which hydrogen cyanide will be released into that particular accidental area. So the route of exposure in that case will be inhalation. Okay, apart when such burning incidents takes place, apart from hydrogen cyanide, there is a possibility of carbon monoxide poisoning also. So if the victim is suffering from both cyanide poisoning as well as carbon monoxide poisoning, no, it becomes much more challenging to treat the person with the nitrite poisoning. Okay, smoke or inhalation victim, as it further reduces the blood oxygen carrying capacity. Okay, this is a typical cyanide antidote kit of nitrite. And you can see amyl nitrite as well as sodium nitrite. They are methaglobin inducers. Methaglobin reversibly binds with cyanide to form cyanomethaglobin, and sodium thiosulfide, it will cause reduction to form thiocyanate, which is excreted renally. This is the important thing you have to remember in your mind. Again, I told you, if you look into the typical mitochondria, which is the powerhouse of the cell, inner membrane space, you have complexes 1, 2, 3, like 1, 2, 3, 4 is there. At complex 4, particular cytochrome CAA, there this particular reaction is taking place. The consecutive complexes 1 to 4 in electron transport chain, cyanide poisoning result in arrest of electron flow to complex 4. Okay, which depends upon heme as a cofactor. Cyanide has high affinity for the Fe ferric iron. Heme A3. You have to remember cytochrome A3 and cytochrome C oxidase forming an adduct, which formation leads to inhibition of this electron transport chain. Okay, that is the reason there will be rapid action. Important organs, brain as well as your heart will be deprived of oxygen. Okay, uh, in addition to that, I have come up across some important pages in the lesson where this cyanide or hydrogen cyanide is mentioned. So we will try to integrate all those pages and read. So please read chapter 3, that is mechanism of toxicity, in which about cyanide is mentioned. Okay, page number 27. Carbon monoxide, cyanide. Other examples are also there: hydrogen sulfide and azide. They are examples of nucleophiles that form coordinate covalent bond with iron. So with this Fe plus 2 at molecular level, what it is forming is coordinate covalent bond, various heme proteins. So this you read. In addition, table 3.2, you read about how the agents which are impacting mitochondrial ATP synthetase. As I told you, mitochondrial ATP transport chain is interrupted, because of which ATP is not formed. So please read chapter 3, mechanism of toxicity, only page number 34, in which they have clearly mentioned. C point is the same: cytochrome oxidase complex 4, cyanide, hydrogen sulfide, azide, and formate. Other examples are also given. So what they are doing, they are inhibiting the electron transport chain. Particularly, if you look into the electron transport, you have different complexes are there. If you see, complex 1 is NADH coenzyme Q reductase. Complex 3 is coenzyme Q cytochrome C reductase. And you have some examples also. See, one question on paraquat you will be getting in your exam, and another this is a experimental molecule MPP. Some questions on MPP also we have come across in DABT. So this also you read. Similarly, you can go through other examples also. For example, phosphine is there. Phosphine is released into your body when phosphorus uh, is there in the acidic pH in your stomach. No, this phosphorus will convert into phosphine, which is much more highly toxic. So, like that, you try to remember the examples. Here we have peroxynitrate C, NOO minus, which is a component in the case of photochemical fog. So, whenever you are preparing for the exam, you try to integrate all this concept. Pause here video, try to remember which particular complex, which type 1 2 3 4, and examples. Okay, so let's see another important page. So I came across another page, that is chapter number 3, biotransformation of xenobiotics, in which they have given whatever this alkyl thiocyanate is there, no, undergoes glutathione conjugation of electrophilic heteroatoms, and their cyanide is getting reduced. So please read about biotransformation of xenobiotics also. Then I came across table number 16.1, in, which is there in the case of toxic responses of nervous system only. Okay, so here you can see which are the chemicals which are associated with neuronal injury. Since we already know major target organs in the case of CNS, this cyanide toxicity is your CNS as well as heart. So coming to neuropathies, how they are doing this, what are the neurological findings you can see? The patient undergoes coma, convulsions, rapid death, delayed Parkinsonism, dystonia. So we try to remember these findings, neurological observations. And what is the cellular basis means? At cellular level you are finding neuronal degeneration, okay, cerebellum, globus pallidus, focal demyelination, block cytochrome oxidase, ATP production. So these are the things which are happening. Okay. Next, in addition, please read table 26.1, in which they have given poisoning syndromes which are associated with or caused by plants. So in chapter 26, you'll come across in the case of toxic effects of plants and animals only. So as I told you, know this cyanide, it is this particular thing is ubiquitous in nature. It is also available present in plants. So there are it is in the form of cyanogenetic glycosides, you can say. What they will do is that to protect these plants from the herbivorous grazing animals, no, they in their kernels, these the small quantity of this cyanogenetic is there, that you can come across in bitter almonds. There are some examples are also given, see, you can go through. And this undergoes gastric hydrolysis, then they will release this cyanide, that thereby it will cause toxicity. So in huge quantity, if you read, as I told you, know already your body will have detoxification mechanism. So take consuming these foods with small quantity, it is not going to cause any toxic effect. But if you're taking in high amounts and your system is already your body is saturated with your detoxification system, there is a possibility of causing such kind of poisoning from eating such contaminated foods. Okay, so also read in under cyanogens. I told you, no, let's quickly read what are the important points that we came across in chapter number 26. Cyanogens are found in wide variety of plants, including kernels of apples, cherries, and peaches. These are the sources. This metabolism of this amygdalin in peaches releases hydrocyanic acid (HCl), and this will bind to Fe+3 iron in the case of methemoglobin and cytochrome oxidase system, which, if severe enough, results in cyanide poisoning with death from asphyxiation, that is difficulty in the breathing, okay, lack of oxygen to the lung tissue. Cassava is another important thing that you can go back and read in African population. So this particular cassava root, which is which is, if it is not treated properly, and if it is consumed in large quantity, there is a possibility of causing this poisoning. So cassava, which is produced by this manihot esculentus euphorbiaceae family, it is a major food source for some regions in the case of Africa. If this particular raw root, it contains cyanogenic glucoside, that is linamarin. So some question you might get about this also, that must be removed during processing of the root. If it is not processed properly and if it is overconsumed by human, then there is a possibility of causing cyanide poisoning. So in addition, I have come across in case of animal toxins only, small quantity it is also found in diplopodia and millipede. You can go back and read page number 394 to chapter number 26. Here you can clearly see that these animals know on the sides of their bodies from this toxins will be released. Okay, in that hydrocyanic acid is also there, and they will use to produce this hydrogen cyanide. So pause the video, try to read about this particular thing, how the symptoms are there. Okay, then, as I told you, no, particularly in the case of African countries, you have come across this cassava. If it is not treated properly, how it is causing. So, first metabolic food reactions we will see that is there in the case of table 3110, page number 459, chapter number 31. It comes under food toxicology. There are various food sources are there like lima beans is there, cassava root, millet that is sorghum, bitter almonds. Okay, that is the reason whenever there is a cyanide poisoning, you will have bitter almond smell or taste, you will have odor, you will be having. Okay, apricot, peach pits. So that particular reaction is called cyanosis, and how the mechanism is this cyanogenic glycoside, they will release this hydrogen cyanide whenever they come in contact with your acidic pH in the stomach. That time only this HCN will be released. That time it might cause some toxicity. So, coming to about order, I told you characteristic orders which are associated with poisoning. Please read in chapter 33, that is clinical toxicology. They have given under, they have listed under table 33.2. They have given whenever there is bitter almond taste, the potential poisoning, they are assuming that it might be cyanide poisoning. Okay. If you have some time, pause the video and try to read about garlic order is there. There is organophosphate poisoning or thallium metal Wintergin order is there, and it is because of methyl salicylate poisoning, right? Then, in continuation of this clinical toxicology only, let's little go through the mechanism. It is mentioned in page number 475. See, various antidotes will activate different mechanisms. Particularly, so how the sodium nitrite is acting is that it, when given to poison patient, forms methemoglobin. This methemoglobin, it is acting as an alternative binding site. So instead of this cyanide going and binding to Fe+2 in hemoglobin, it will go and bind Fe+3 methemoglobin. Okay, thereby it causes less toxicity in the body. So in that only, I have come across this particular in at the end, no, you have at the end of the chapter you have some questions will be there. So one question was there in the textbook only: which of the following might be used as an antidote for treatment of patients with cyanide poisoning? The right answer will be sodium nitrite. So pause here, try to read epicac, how it is used. Maybe it used as an as a, for vomiting, for inducing vomiting. Atropine in the case of maybe the muscarinic cholinergic type of poisoning, atropine act as an antidote. Chelating agents, what are those chelating agents? The bal, dimercaprol, all these things are that also you read, and quinine also. So try it, not just stick to one option, try to read other options. What are the, in what type of poisoning these are used as an antidote. Okay. So, I think I have come across in chapter number 34, that is in occupational toxicology also. Please read this table, table 34 2. They have given some examples of occupational diseases and the toxicants that cause them. In that organ system, lung and airway, they have listed that hydrogen cyanide is the one which is causing asphyxiation. So pause the video here and try to read about other things also that you can remember with respect to causative agent and diseases. For example, phosgene, how it is causing pulmonary edema and bronchiolitis. Okay, carbon monoxide is also causing asphyxiation. Cotton dust, chronic bronchitis. Okay, silica asbestosis, fibrotic lung tissues. With respect to occupational disease, one question you will definitely come across. For example, hypersensitive pneumonitis, which is created by these bird proteins, pyrethrum, penicillium, aspergillus, metal fume fever. Again, one of the frequently asked topic with respect to which is caused by either zinc, copper, and magnesium. Those workers who are working in this industry. So please go in detail, read about metal fume fever. In my channel also, one video I made on this also. Like this, pause here, try to read this table, try to remember the examples and what kind of occupational disease it is causing. In addition, I have also come across that there are, if you look into the historical, there is a cult, a mass poisoning is there with respect to in African country you can come you have seen that this particular is called Jonestown mass suicide is there. In this there was a gathering of hundreds of people were there. I think close to more than 800 900 are there were there where they have been consumed with a juice which was contaminated with which was poisoned with a cyanide. So this is a very famous historical incident is there. Please go back and read about Jonestown cult mass suicide. And also historically if you see Zyklon B, another important thing which is also commonly known as gift gas. This particular cyanide, no, um, hydrogen cyanide, it was used in the case of Nazi concentration camps. Okay, so go back and read about this thing is which was used as a. There are a lot of other historical incidents where you have seen that cyanide is used as a suicidal agent, homicidal agent also you can say. So till now, we have discussed nitrite, their mechanism of action. I mentioned about sodium thiosulfate also, which will try to convert it to sodium thiocyanide, which can be excreted renally. Another important is vitamin B12, that is cyanocobalamin. It has a greater advantages when compared to nitrite. Question is not related to this one, but I'm bringing this third one also, so that any question comes with respect to this also will be able to answer in the exam. So important point you have to remember: hydroxocobalamin when combined with cyanide, it will produce cyanocobalamin. This particular cyanocobalamin, it is very inert and it can be excreted through kidney. This hydroxycobalamin causes an increase in blood pressure. So whatever the challenges which we had with respect to nitrite, which causing hypotension, no, that can be overcome by using the cyanocobalamin, which can be desirable in the case of cyanide poisoning. And only disadvantage is that the patient will show red discoloration. This red discoloration of the skin as well as body fluids. Urine becomes red. But don't get panic. It's a well-known effect that once you administered this particular cobalamin, there will be change in the skin color as well as the body fluid. Hydroxycobalamin does not pose a risk to the patient with concomitant carbon monoxide. Another important thing, if the patient the exposure is through the burn, through the fire accident, and there is a mixture exposure of both cyanide as well carbon monoxide. In that case, drug of choice will be cobalamin. Exposure the same way as the older cyanide antidote. That is, older one it used to contain amyl nitrite, sodium nitrite, and sodium thiosulfate. The older cyanide antidote kit induces methemoglobinia, which when compounded with significant carbon monoxide exposure would pose significant limitation of oxygenation of the patient. So that is the important thing that you have to remember. So hope from this video you were able to understand the importance of cyanide poisoning, the mechanism, three different types of treatment, and the sources of exposure, ubiquitous nature. I've given out the important pages also in the textbook where from where there is a possibility of asking a question. Okay. Thank you so much. Be curious. For any questions, you can always write a mail to arya.priyanka18@gmail.com. Like, subscribe, and follow my YouTube channel, Toxicology on Board. All the best for your exam.